All right, we are back again talking about our series, What is Truth? Once again, there's the first 5 that we've gone through, and we are still on number 6, as we will be for a while, that truth doesn't equal science, and science doesn't equal truth. So the past 2 weeks I gave you an overview of the structure of DNA, and I gave you evidence found within that structure, yes? We're actually going to get to that. How do you define science? We're going to get to that. Okay?
I don't want to backtrack, but
We will get to that. Be patient. And last week I introduced what's called the design inference. And as I said, the features of logic and reasoning involved in the detection of intelligence and design come from a book called The Design Inference, written by Dr. William A. Dembski. And as he writes, and as I said last week, the presence of an improbable object and the presence of patterns are the 2 criteria that indicate both prescience and intelligence were used in its creation. And those 2 things infer design. Yes? Absolutely. That is correct. It's sad that today it's become like a battle between science and God, but actually authentic science actually supports and reveals things out of the Bible. That is correct. All right. So, so we have design that is inferred and the intelligence and the creation of that design. So when you have a designer, or a design, it also means there's a designer, because it doesn't come from randomness. When you have patterns in an improbable object, like you do in DNA, we therefore can state that it meets the criteria for being designed. So whenever we encounter information-rich systems, for example, an ATM, a pyramid, a monument, a language, a car, a gas pump, Stonehenge, the Great Wall of China, which we brought up last week, right? Computers, software, airplanes, radios, maps, watches. We could sit here and do this all day. Anything, any one of those types of things, any information containing system that we're bombarded with on a daily basis now, when we have those, we recognize the improbability of their being created by natural forces or randomness, and the recognizable patterns that they contain, and therefore we know they have been intelligently designed. And we know this because intelligence is a requirement for the design and creation of information-rich systems. Intelligence produces information-rich systems, not naturalism or randomness. So, has everybody seen the original 1996 movie, Independence Day?
yeah.
Okay. In that movie, the opening scene depicts the detection of an electromagnetic radiation signal in a lab involved in the search for extraterrestrial intelligence, or what they call SETI. Now, although that signal and the remainder of the movie are fictional, SETI labs are not. SETI labs monitor electromagnetic radiation entering our atmosphere for signs of transmission from intelligent civilizations on other planets. They do this in an attempt to find radio signals containing information-rich patterns. So that begs the question, why do they do this? And why are they specifically looking for signals with information-rich patterns? And the answer is, if they ever detect one, they know it will signify the presence of intelligent life beyond Earth. They know.
I'm sure that they've already gotten what they wanted.
They know that a signal with a pattern indicates the presence of intelligence in its creation, because such information could not have been generated as a result of randomness. And I think it's funny how they recognize this with ET, but not with our own planet. It's kind of ironic, huh? They recognize this with ET, extraterrestrials, but they don't recognize it right here on our own planet. This, ladies and gentlemen, is the design inference once again. Again, intelligence produces information-rich systems, not naturalism or randomness. So whenever you encounter an information-rich system, you know it infers design, not randomness. And this holds true for the natural world, not just for man-made objects. And if you want to talk about information-rich systems, there isn't a single system in existence, man-made or naturally occurring, that is more information-rich than DNA. None. Period. And if you try to explain the existence of DNA, you not only have to explain its structure and its functions, you also have to explain the information that it contains. See, it's not only DNA's structure and functions that are unbelievably unique, complex, organized, choreographed, whatever you want to call it, controlled, structured, patterned. It's also the information that it contains. And you need to get this, please. DNA is a biomolecule that stores information, a massive amount of information that's incredibly complex, logical, organized, and patterned. Therefore, if evolution could account for DNA, it would not only have to account for the molecule itself, but for the origin of the mind-boggling amount of information that it contains. It not only contains, but it stores and utilizes in order for life to happen. Any attempt to explain the origin of life is absolutely required to explain both of these things about DNA. This is non-negotiable. However, sadly for evolution, it has no answer for either of these. It can't explain the molecule itself, and it cannot explain the information that DNA contains. That it not only contains, but it processes in a manner far superior than anything we've ever invented or ever will invent. Nothing in mainstream secular science has ever explained the origin of DNA, much less the information that it contains. Dr. Werner Gitt says this: there is no known law of nature, no known process, and no known sequence of events which can cause information to originate by itself in matter. So to give you some examples, or a perspective maybe, on the amount of information that DNA contains. When I look at the character and page counts of my manuscripts, what you end up with is that an average 6x9 book, okay, printed in a typical 11 or 12 point font, would be about 150 pages long, okay? Those 150 pages would contain approximately 400,000 characters, including the spaces. So, with that said, if you printed the entire sequence of one strand of human DNA, 3.2 times 10 to the 9th, 3.2 billion bases. Or, if we're talking page count, 3.2 billion characters. Without spaces, it would fill about 8,000 of those 150-page 6x9 books. Now, if you want to print the other strand, now we double that, obviously, and we're up to about 16,000 of these typical sized books. But here's the thing. The information DNA has isn't just the sequence of its bases. To give you an example of how much information is contained in DNA, I was taught that if the information from DNA from one person was typed out, it would fill enough books to fill the Grand Canyon approximately 40 times. Okay? From this, you can clearly argue that DNA contains more information than all the books in all the libraries in all the world. Geneticist Michael Denton describes it this way. The information needed to build the proteins for all the species of organisms that have ever lived, a number estimated to be approximately 1,000 million, could be held in a teaspoon, and there would still be enough room left for all the information in every book ever written. Ponder that for a while. It is mind-boggling when you think about that. It is simply amazing. It's like nothing else in the world. But it's not just a massive amount of information. The information it has is vastly superior to anything we have ever created or encountered. Bill Gates said it this way. DNA is like a computer program, only much more complex than any we've ever come up with. And speaking of DNA to a computer, the amount of information stored in 1 cubic millimeter of DNA has been estimated to be equal to 1.9 times 10 to the 18th bits. That's 1.9 quintillion bits. Folks, that's a big number. As Bill Gates says, this is vastly superior by many orders of magnitude than any of the greatest machines we've ever come up with or ever will come up with. It is truly amazing. And then when you think about the way the information is created, it's so simple. Remember, 4 building blocks. That's it. Creating all this information. It is truly mind-boggling. But it doesn't just store information, it uses it to create, develop, grow, sustain, reproduce, protect, and heal life itself. Yes, sir?
Is all this information, like you said, it's made with 4 components, like, is it the way that it's set up that makes the information, like each strand of DNA is made up a certain way?
That is absolutely correct. It's the order that those 4 bases recur within the DNA.
You have the father and the mother, these two, they both have like a certain strand of DNA that's for hair color. So both of these things are compatible, but when they come together, they'll come together in a different order. But
that is correct.
in order to get that.
That is correct. It then ends up becoming unique for each offspring because half's coming from your mother and half from your father. Yeah. Mm-hmm. Yep. Okay. So without DNA, life as we know it would be impossible because it does all these things. It contains the information needed to recognize and repair damage and errors within itself, to reproduce itself, as well as knowing when it needs to reproduce itself for normal cells, somatic cells, versus germline cells for reproductive cells. It also has all the information needed to regulate itself, to protect itself, to store itself, to turn itself off and on in different areas in the genes, etc., on and on and on. And it does all this through its 4 basic functions, and that's what we're going to discuss now. And when you talk about these, you usually go right here to what's called the dogma of molecular biology. If you look at this, DNA is used through the process of transcription to make RNA. RNA is then used through the process of translation to make proteins, and DNA is used to make more copies of itself through the process of replication. And there's a fourth function, not shown here, which we're going to talk about, which is the recognition and repair of damage and errors. And in these four functions, there is an overwhelming amount of scientific evidence that shows evolution is not possible scientifically. So that's what we're going to do. And what I want to start off doing is first briefly explaining these four functions. And we're going to start with the first one up there, transcription, where DNA is used to make an RNA copy of itself. And this creates an RNA molecule that's complementary to a DNA sequence. Here's a graphic for that, a cartoon. RNA is a nucleic acid just like DNA, and it's also made up of four building blocks. But the RNA nucleotide building blocks are a little different than DNA's. And let me show you this. If you remember, DNA gets its name from where we're showing here on this arrow, the deoxy 2' position. It's missing, so to speak, an oxygen there and only has a hydrogen. So it's deoxy. Hence the name, if you remember, that's why they call it deoxyribonucleic acid. Well, RNA has a more common version of that. It has both an oxygen and a hydrogen at the 2' position in the ribose sugar. So it has oxygen, so you don't need to call it deoxy. So RNA then gets its name from there. It's just ribonucleic acid. You drop the deoxy because it has an oxygen there. RNA is also usually single-stranded as opposed to DNA being double-stranded. Now RNA, like I said, has four building blocks like DNA, but they're not the same. Three of them are A, G, and C, there we go, are the same. However, where DNA has a thymine, a T, RNA uses uracil, which is a U. So three of the four are similar bases, are the same base type. One of them is different. So back to this, our little graphic. So what happens here is, this is primarily accomplished through a protein called RNA polymerase, which is our oval there, the RNA pol. It runs along, reads along the DNA strand after it's opened up, and what it does is assembles the RNA, which I'm showing in red. One nucleotide at a time, each line on there represents a new nucleotide. And all of those base pairs, or all of those nucleotides from RNA, are ones that can base pair with that DNA strand that it's going along. So as it goes along there, RNA polymerase matches an RNA A with a DNA T, RNA U with a DNA A, RNA G with a DNA C, RNA C with a DNA G. Notice the pattern of bonding is still the same. G with C, A with T. However, since RNA doesn't have T, every place where you would have a T, RNA uses a U, and that binds with the DNA A. So it runs along there, reads the DNA, creates an RNA molecule by adding one nucleotide at a time that is complementary to or can base pair with the DNA that it reads. So when it's done, the RNA molecule, if the DNA stayed open, would be able to base pair right in that same section of which it was just made. Now, if you're paying attention here, if you remember We have 3.2 billion base pairs of DNA divided into 23 chromosomes. So how in the world does RNA polymerase know where to start reading the DNA and where to stop? Because no matter where you start, if you start at a different place, you're going to get a different sequence because it's going to begin elsewhere. So here's how this happens, and this is where the term gene comes into expression, or comes into our discussion. Jeans, Levi's, Lee's, Wranglers, whatever you wear, right? Jeans. Wish it were that easy. However, these genes are much different. A gene is defined as a unit of heredity, which encodes a functional RNA molecule, contains all the regulatory aspects to ensure that the RNA is transcribed accurately. So it's a stretch of DNA, okay, that's used as a template to make an RNA, which is then typically made into protein. When you draw genes, they usually do something like this. Just a very simple cartoon, but each different region is indicating what is happening in that stretch of DNA. But again, all it is a stretch of DNA. But it contains both the bases that are used as a template to make the RNA and all the regulatory regions that tell the RNA polymerase where to start and where to stop and things like that. So you could say it has the functional bits. And let's use this analogy, like the street itself. And all the regulatory bits, like the stop signs, the stop lights, the street signs that tell the cars, the RNA polymerase, where to start, where to stop, which way to go. So a gene contains all of those things. After RNA is made, it undergoes some processing. Some bits are chopped out. That's what they call introns mostly. That's what most of those are. And some are kept. That's what they call exons. That's the part that ends up in the final RNA product. And when this is all done, the final RNA molecule is called an mRNA, which stands for messenger RNA. And then it gets passed into the cytoplasm, where our next DNA function comes up, which is translation. This is the process of creating proteins from the mRNA. Through translation, just like it sounds like with a language, the language of DNA, which has been copied to RNA, which is bases, gets translated into the language of proteins, which is written in amino acids. So through translation, we go from the building blocks of DNA and RNA to the building blocks of proteins. And that's primarily accomplished through a structure called a ribosome. They are the Rosetta Stones of what's called the genetic code. In a minute, I'm going to show you that. So they translate the information from the bases into amino acids, which are the building blocks of proteins. And there are 20 essential amino acids. And the secret behind doing all this for the ribosomes is found in a word called a codon, which means it does everything 3 bases at a time. Here's an illustration of that. A lot going on here. So let me explain this to you. The red down there, that's our mRNA. The sort of shape that sort of looks like a top hat in the back, that's the ribosome, okay? So that's the setup for that. It functions kind of like a conveyor belt. It runs along the mRNA, reading 3 bases at a time. Each codon, each triplet of bases, has a corresponding amino acid that it codes for. So it's kind of like using your secret decoder ring from Captain Crunch, right? Okay? As it's doing this, it's going to read 3 bases, And then it's going to look for a molecule called a transfer RNA, or what's called a tRNA. tRNAs have amino acids attached to them. So when it reads those 3 bases, it's going to grab the tRNA that binds to those 3 bases. And as it does, you see what happens is it brings its amino acid with it. So now that we have it there, it matches the codon with the tRNA 3 bases. And now, once it's matched up, the ribosome removes the amino acid from the tRNA and starts making a long strand of it by binding it to the previous one. So if you look in there, the circles I'm showing are the amino acids being added one at a time to the pre-existing string of amino acids. It does this, goes all along this, till it gets to the mRNA at a point where it reaches a stop sign. There are codons that also tell it where to stop, just like there are ones that tell it where to start. In doing so, they use what's called the genetic code. This is the genetic code. It's showing all the 3 base codon possibilities and the amino acid that associates with those 3 codons. And notice there's also 3 that are stop signs. And the start sign isn't labeled as start. It's AUG down there, methionine. That's our start sign. So it has all the codons it needs, finds the tRNA that matches it, and then this will be the amino acid that's called for, goes in the chain next from that codon. After it gets to a stop sign, you'll have this long strand of amino acids. Then they need to get folded into its final 3D structure, because a protein is absolutely useless until it gets folded into its 3D structure. So there are proteins that do this called chaperonins. They fold it up then into its 3D structure. And this is critical because proteins do the many life-essential jobs of our cells, our tissues, our organs, and our bodies. So just like DNA and RNA, they are essential for life. So that brings us to replication. This is the process where the DNA molecule is copied. So replication is like our Xerox machine. We're going to make a copy of it exactly what it was like in the first place. It is essential for this to happen, for life to exist, and for life to be passed on. Because when a cell divides, you got to give it the same DNA that the original cell had. So when a cell divides, it's got to have an extra copy of DNA to be passed on to the daughter cells. So replication is absolutely critical for life. And we have, like I said, 3.2 billion base pairs of DNA packed into and condensed into our 23 chromosome pairs. All of those have to be replicated and evenly distributed to the daughter cells each time a cell divides. There's 2 different ways that happens. Typical cell division, mitosis, or cell division when you're making the germline cells, the sex cells, that's called meiosis. So sometimes when you do meiosis, that is an example where the copies of DNA are not evenly distributed. However, that's a whole nother topic. And we're not going to get into that. We're just talking about how DNA gets copied. But just so you know that sometimes the DNA isn't evenly distributed when you're making the sex cells. All right? But we're just sticking to how the DNA itself is copied through replication. And this is primarily done through an enzyme called DNA polymerase. It creates 2 new strands of DNA using the existing strands as its template. Here's an illustration of that. And if you look at that, you would say there is an awful lot going on here. And that's kind of the point. And what I'm just showing you is the major players. The ovals, triangles, etc. There's our DNA pol. That's our DNA polymerase, the major one. Like RNA polymerase, DNA polymerase runs along the DNA strand, reads the existing strand, and grabs a DNA nucleotide that base pairs with the existing nucleotide, adds it to the growing DNA strand one at a time. And this happens at things called replication forks. That's why I'm showing it shaped that way. That's a replication fork. And basically what's happening here is that numerous proteins are unwinding and separating the two DNA strands, relaxing it from its supercoiled nature, holding it open so DNA polymerase can get in there and run along it, and then closing it back up again once DNA polymerase has copied the DNA and made two new strands. If you look at the bottom, the replication direction is one direction. However, if you look at the two strands on top, they're going in different ways. So the actual copying of the DNA is bidirectional, but overall, it's only going one direction. This gets rather complicated when you study how this happens, but there are more proteins involved in doing this. Suffice to say, when you're done, you end up with two identical copies of the DNA molecule. That's the bottom line. Yes, sir?
Okay, I have a question. You know, test tube babies and stuff?
Uh-huh.
Is it true that you can manipulate the DNA to, get, certain, results? Yeah, you want, blue eyes and stuff for the baby, then you can manipulate the DNA so it replicates the...
We are not at a point yet scientifically where that is something that can be standard done. That that just it is scientifically possible, but it involves a lot of work and a number of risks. But the science to do things like that is there. Whether or not it's ethical is a whole nother topic, right? We can talk about that in our small group later on, okay? Now, the last thing I want to discuss briefly is the recognition and repair of damage and errors. DNA can be damaged chemically or environmentally, and errors can occur during transcription and translation. Transcription and replication. These things can all cause strand breakage or the improper pairing of bases. But DNA codes for proteins that recognize and repair damage and errors. And in many cases, it's the same proteins involved in replication that are recruited to do this type of work. Whenever errors and damage occur and go undetected or unresolved, it results in what are called mutations. Mutations are alterations in the normal DNA sequence. They can be as small as one single base pair, or they can be as large as an entire chromosome. They can be the deletion of DNA, or the insertion of new DNA, or a change in DNA. But here's the good news. Mutations are very rare, and they rarely have an impact on the individual. That's because DNA does an outstanding job of making sure that everything that happens in these processes happens correctly. Okay? So with that, I'm going to end the discussion of the background of these processes. And so I just went through entire college courses in about 20 minutes. All right? Next week, we're going to discuss evidence found in these functions, and most of that is going to be next week. But I want to end today giving you one bit of evidence, and then we'll pick it up next week with more from that point. And to discuss that, I'm going to go back to this slide, the dogma of molecular biology. So the one thing that I want you to get from our discussion of these processes today is that they are amazingly complex, choreographed, intricate, detailed. These functions, sorry, wait a minute, are amazing enough on their own. But what's even more information, or what's more amazing, is that the information, that's what I meant to say, needed to make these processes happen, all comes from DNA. So we need to advance this slide, which shows our processes, to this slide. Because this now becomes the all-time greatest catch-22. Look at this. I'll explain this. You'll see. You need the RNA and the proteins to make the DNA. But you also need the DNA to make the RNA and the proteins. Not only that, you need pre-existing DNA to create DNA. So the question is, how in the world can this happen gradually and randomly? And the answer to that is, it can't. It is not possible. They must all be there, existing at the same time, functioning together, or none of this can happen. And if none of this can happen, then life can't happen. Furthermore, these processes that we just went through absolutely require at least 100 unique proteins and about 200 unique RNA molecules. Because you need about 100 RNA molecules to create the 100 proteins, and you need more RNA molecules that function just as RNA molecules. So it ends up being about 300 molecules that are absolutely required for all these life-essential functions of DNA. But you can't have any of these 300 molecules without the DNA. And you can't have the DNA without any of these 300 molecules. You need the DNA to make all these 300 molecules. But you also need these 300 molecules to make the DNA. And not only that, as I said, you need pre-existing DNA in order to make more DNA through replication. Folks, this is science. This is what everybody knows. Therefore, each one of these 300 proteins and RNA molecules must all be present at the same time from the beginning, or you cannot have life. But it doesn't stop just at their presence. As we were learning, all these types of molecules interact with one another, and they must function together. They must all have the appropriate and necessary subunits to be able to interact with one another. And so they must know the function and subunits of the other molecules with which they must interact. So here's the question. How can all these molecules arise gradually, randomly, at various times, independently of one another, and yet still have the structures required to interact with one another, as well as those needed to interact with DNA? Furthermore, how do all these molecules end up being encoded by genes in the DNA if they came into existence gradually and over long periods of time? What I hope you see is all this represents an insurmountable problem for evolution. It's insurmountable because it simply cannot happen evolutionarily. And all of this is what is referred to as irreducible complexity. Irreducible complexity means All the parts must be present together at the same time, functioning in concert with one another, or the system as a whole cannot and will not function. All the parts, subunits, components are all needed in their entirety at the same time in order for the system to function and function properly. They're all equally necessary, so they all must arise and be present at the same time for the system to function. You cannot develop the system gradually because all the parts have to be there together simultaneously. Think of it this way. A car without a transmission does no good. All the parts have to be there together at the same time. So hopefully what you can see is that irreducible complexity is a death sentence for evolution. And that's where we're going to leave it today. But we're going to pick it up next week and talk more about this topic as well as more evidence that we find when we look at these 4 functions of DNA in molecular biology.